Thank you for kind introduction, Honma Sensei. So I would like to thank you for giving me opportunity to speak here today. And I'm sorry I could not join you in person, but I am very happy to share our research on genomic medicine. So Honma Sensei already introduced about my works, but during my PhD program at Kyoto University, I was working on time series analysis of protein dynamics using molecular dynamics simulation. Then when I was assistant professor at Keio University, I was working on the genome analysis of microorganisms, especially for natto in Japan. So that is the start of my genomic analysis research. And since 2015, I have been doing research about genomic medicine at Kyoto University and the Kyoto University Hospital. And in our laboratory, biomedical data intelligence, together with Professor Yasushi Okuno, we are working on the research for personalized and precision medicine and innovative drug discovery. Now, I am mainly working on the research about genomic medicine and computer simulation for genomic drug discovery. So today I'd like to introduce our work related to these topics. I have put protein structural dynamics in my talk title, but I would also like to introduce research other than simulation. So I think many of you know what is genomic medicine, but genomic medicine is the practice of providing accurate diagnosis and appropriate treatment based on individual genetics differences. The practical application of genomic medicine have already begun, especially in the field of cancer. In cancer, some genes are activated or inactivated by genetic variation, and the molecular targeted drugs for the gene have been developed. So based on the molecular profile, in this case, genomic variation pattern, patients can be stratified, and we are able to establish a treatment plan for each subgroup. In Japan, cancer gene panel testing is being implemented under the National Health Insurance System from 2019. It shows a rough flow of clinical sequence for genomic medicine. Genetic variation are detected from sequence data of patient sample. Then detected variants are annotated and curated using the public databases. Then the report of clinical interpretation is made, and physicians use this to decide diagnosis and treatment policy. The clinical interpretation for variants is currently being done manually based on the information from databases and publications. There are some guidelines to define the clinical significance of variants. These guidelines define how much the information can be used as evidence to define clinical significance, and they state that information about genetic variation that can be obtained from public database is important evidence for clinical interpretation. So there are many databases has been developed, and especially ClinVar, developed by NIH, is often used in clinical interpretation. Its database provides relationship between genomic variation and disease. For somatic cancer, there are also some databases that provide information about variants with effective drugs and also evidence. The information in these databases is important and very useful for clinical interpretation. On the other hand, recently it has been pointed out that genomic data accumulated in public databases have a population bias. This paper investigated the ESIC group from which the accumulated data were obtained for the GBAS catalog. It is one of the popular database in our field. And they report that more than half of the genomic data are from European population in this database. So as you know, there are differences on genomic background between population, and these differences affect on efficacy of drug and risk of onset. And there are also reports that such data bias can also affect the result of genomic medicine. This pie chart shows the result of clinical interpretation for variants detected by genetic test by the ESIC population. So here, VUS, it indicates that clinical significance could not be assigned. So it can be seen that the ratio of VUS in Asian is higher than Caucasian population. So we need to accumulate genomic data in each population, for us in the East Asian population, and we need to increase the diversity of the data. So AMED, which is a Japanese founding agency for medical research and development, has started a program for integrated database. In this program, first, the research groups have been adopted by AMED program in the field of cancer, rare disease, and dementia, and infectious disease, and sensor disorder. They recruit patients, perform sequence analysis, collect genomic data and clinical information, and then share these data with the group. And then these data are integrated into an integrated database. So we developed this integrated database. So this database provides disease-related variant information and basic patient information without any restriction. So the database is named MGen. So we published this database in March 2018. So I hope you will be able to access it from this URL or this QR code. So MGen is the first database that provides disease-related genomic information specified in Asian population. And this database also integrates variant information detected in not only monogenic, but also polygenic disease. You can use MGen by searching gene name, variant, and position and disease name from this top page. And here is a search result. The MGen provides original annotation from our submitters and also information by ClinVar and other databases. And in the variant detail page, you can see the detailed information registered in MGen along with the basic information about the variant. The frequency information for age and gender for each variant, as well as information about the submitters are also available in this page. And here is the number of variants published in our database. So it is not the latest one, but more than 100 unique variants are published in MGen, and 74% of those are only in MGen, not only ClinVar. So as you can see, the effort to collect genomic information by each ESIC group, each population is very important. On the other hand, there is another problem. Many variants have not been annotated with any clinical interpretation because no annotation has been reported for each variant, and its functional role has not been experimentally determined. So these variants called are called VUS, variants of uncertain significance. Actually, in this paper, sorry, maybe it's too small to see, but each line indicates the performance of cancer gene panel testing. In some panel, for example, here and here, they report more than 40% of detected variants are annotated as VUS. For these variants, we cannot take any clinical action. So it is said that VUS is bottleneck for genomic medicine. So to support clinical interpretation of VUS, we have been developing some in silico approaches. First, we developed a machine learning model to predict pathogenicity of variants. The second, we estimate the functional effect and differences on drug efficacy by variants using molecular dynamics simulation with supercomputer FLOC. So first, machine learning about machine learning approach. There are many computational methods to predict pathogenicity of variants. Actually, the guideline, as I introduced in the previous slide, they recommend using computational prediction score to support clinical interpretation. The conventional approach uses information from amino acid or nucleic acid sequence information and protein structure data. Using them as feature vector and input for machine learning model, then calculate the probability of pathogenicity of each variant. So there are ensemble methods that use predicted score by informatics tool as input feature vectors. They improve accuracy by comparing with the accuracy using each tool only. Here, in fact, in living organisms, molecules interact with each other, and these relationships are also involved in the mechanism of disease onset. However, existing methods don't consider molecular interaction on the prediction of pathogenicity. So we have developed a model to predict pathogenicity using molecular interaction information. And the model name is PathoGN. Here is a flow of existing methods, as I showed in the previous slide. In our method, first we construct a graph representing molecular network, then integrate predicted score from other bioinformatics tool, and together with variants information from public databases. Then construct a knowledge graph like this. Then using neural network model, predict the pathogenicity of each variant. To construct knowledge graph, first we use Reactome database to construct the molecular network graph. And variance level and prediction score are obtained from Bench dataset. They consist of five benchmark datasets, and they also provide predicted score by informatics tools listed here. So using them, we construct a graph for each benchmark dataset, and then use this graph as input for machine learning model to predict pathogenicity of variants. So we employed graph convolutional network. GCN is convolutional method on graph proposed by Gibbs and Wayne. Here is an example. In image data, CNN, convolutional neural network, very famous model for image analysis. So CNN convolves information from neighboring, in this case, 8 directions. And GCN and GNN convolves the information from neighboring node of the target. And here is our model architecture. So I think I don't have enough time to explain the detail, so I'd like to skip this, but we implemented our model based on KGCN, developed by our colleague Ryosuke Kojima. So architecture is like this. So using this model, we evaluate our model, and here is a result of benchmark validation. I saw the results of two benchmark datasets named SwissBar and ValiBench. In both, our method pathogen is indicated by orange color here. And as you can see, our model outperforms other tools and algorithms. So we also validate the performance of our model with real-world data in ClinVar. In ClinVar, there are variants whose clinical interpretation was unknown in the past, in this case 2019, but whose pathogenicity has become clear with the new evidence now. So we use the new label as correct answer and validate our model using these variants. And here are the results with 2019 and 18 and 70 dataset. For all dataset, our model could estimate the correct label on 2020 with more than accuracy 0.8 and AUC 0.9. So we also predict the pathogenicity for the latest ClinVar dataset, and we will publish them on the web. Now we are also currently working on improving the model to predict the variants especially for somatic cancer. And next, I would briefly introduce the second topics about the functional estimation with molecular dynamics. So why we use molecular dynamics simulation is protein have dynamics within our body. So, and this dynamics plays an important role for molecular function. Using molecular, as you know, molecular dynamics simulation can capture this complex dynamics of protein, and it can be verified at the genomic level. So we have been working on the research using MP-Café method, which utilizes MD simulation to calculate and energetically evaluate the affinity between molecules and compound. In the previous works by my colleague, MP-Café approach have been shown to be useful in some protein system. So we have been using this method to predict the drug affinity of mutated protein. In the collaboration with Keio University, we successfully evaluated the drug sensitivity of EGFR mutation with high reproducibility using MP-Café approach. So horizontal axis is calculated affinity by MP-Café, and vertical axis is observed affinity by experimentally. So each dot represents mutated protein. So based on these successes, we decided to run the calculation on more mutants with supercomputer Fugaku. So now we are working on the project with Fugaku in this project. In this project, we use Fugaku to clarify the effect of genetic variant on protein structure and dynamics. Target variants are patient-derived variants accumulated in our database MGen and also ClinVar and Cosmic. Now we are currently working on a calculation of these variants using Fugaku. We are also accumulating the calculated results by Fugaku, and now we are developing a database to publish and provide the result. So we will probably be able to publish this database in the next few months, I hope. So I am so happy if you could check this database. So here is a summary. Today, I introduced the challenges in genomic medicine and our research on them. In the future, I would like to work on data integration and the development of a method to approach the mechanism of disease onset caused by genetic variations. So here is acknowledgement. Thank you for your kind attention.